glucose catabolism and ATP synthesis. Remember, catabolism, breaking down ATP, the gas of the cell, adenosine triphosphate. So glucose catabolism, reactions that involve the breakdown of the monosaccharide, remember, single chain monosaccharide glucose, and using the chemical energy in the bonds of the glucose to drive ATP synthesis. So breaking down glucose is we break it apart, catabolism, release the energy, use that exergonic reaction to power an endergonic reaction that absorbs energy and create ATP. That's the general idea. So glycolysis happens in the cytosol, splits glucose, and then we have the citric acid cycle, a series of reactions in the mitochondria that continues the process. Oxidative, remember, to oxidize, lose electrons, oxidative phosphorylation, add a phosphorus, phosphorylation, series of oxidation and reduction reactions, remember, losing electrons, gaining electrons, take place in the mitochondria, allows cell to use energy from the glucose catabolism, from the breaking apart of the glucose. And then we have what we call the electron transport chain, which also is part of the oxidative phosphorylation process, which leads to ATP synthesis, transferring electrons, movement of electrons, energy, work. All right, so glycolysis, the breaking apart of the glucose, happens in the cytoplasm, cytosol. Inside the mitochondria, we have the citric acid cycle to produce ATP, and also electron transport chain to produce ATP. This is glucose catabolism, breaking down a glucose, first two steps, and then oxidative phosphorylation. We also have what we call substrate. The ATP can be synthesized in two ways. One of those is substrate level phosphorylation, transfer of the phosphate group directly. And then we have oxidative phosphorylation, which is movement of electrons. Energy flow of electrons is harnessed to produce ATP. So these are two ways ATP can be made. Remember, the energy, once we have ATP, we can use it in two ways. We can use it in a phosphorylation, transferring the phosphorus, or we can use it in breaking apart that last bond and releasing that energy and using it that way. So the oxidative phosphorylation, these reactions generate a great deal more ATP than glycolysis, which is just the breaking apart of the glucose, and occur in the mitochondria as part of the electron transport chain. As we talked about before, the glycolysis and the citric acid cycle means they add electrons, reduce electron carriers, which feed into the electron transport chain. So the glucose catabolism can be divided into two broad classes, the glycolytic or anaerobic catabolism. Anaerobic means not aerobic, means absence of oxygen. So these are the reactions that take place in the absence of oxygen, which involves glycolysis, breaking apart of glucose. So part of it is anaerobic, and then you have the aerobic catabolism. This part requires oxygen. The electron transport chain and the citric acid cycle are aerobic catabolism, require oxygen. And where do you think that oxygen comes from? We talked about that in the respiratory chapter. Right, so the cell consumes oxygen to be able to create ATP. And we also produce carbon dioxide and water. All right, so let's go through glucose catabolism. So glucose is broken down a series of 10 enzyme orchestrated reactions. Enzymes, remember, those are the catalysts to your body's reactions. The catalyst of your body reactions. They help speed up reactions by reducing the activation energy. Glycolysis takes place in the cytosol of all cells. Remember, all cells need energy. It's part of being alive. One of the traits of living is that you need to use energy. So glycolysis, glucose, lysis. So molecule of glucose is split into 3 carbon sugar molecules. Remember, glucose is 6 carbon. You split it into two 3 carbons. So basically split it in half, so to speak. And 2 molecules of pyruvate. And there's 2 phases of that: energy investment phase and energy payoff phase. Right? You're going to see some similarities between these different cycles. All right, so energy investment phase requires energy. And you don't need to know all the steps, but we're just going to go through them briefly here. Right, glucose is phosphorylated, add a phosphorus. Then you have another phosphorylation, another by ATP. So ATP adds a phosphorus group one time, then a second time. Then you have this cleavage. 6 carbon fructose is split into 2 3 carbon molecules. And at the conclusion of this phase, you've had 2 ATP, which have donated their phosphorus to the glucose, and you have the split. And so you've used 2 ATP in this process. And a lot of times it's easier to see it in a diagram. So you have ATP phosphorylates, adds a phosphorus group to the 6 carbon glucose. Remember, the carbons are in a chain structure, in a hexagon structure, but we just put it as 6 carbons side by side for now. And remember that they're not showing the hydrogens that are in this remember, glucose, each one is connected to another, each carbon is connected to another carbon, and 2 hydrogen off of each one. All right, so you use 1 ATP at a phosphorus group. Use another ATP at another phosphorus group, and then you split the glucose. Then you have energy payoff phase, oxidation. Right, so glyceraldehyde 3-phosphate is phosphorylated and oxidized by NAD to yield NADH. And the other one is, and the 1,3 bisphosphoglycerate donates a phosphorus to ADP, producing ATP. ATP synthesis, carbon atoms in 3-phosphoglycerate are rearranged to form phosphophenol. Pyruvate donates a phosphorus to an ADP, yielding ATP. Right, so those sound like big words, but basically what we have is the phosphorus plus the NAD, the reaction plus yield, and then you have an ADP, which will be phosphorylated to ATP. This is going to be an energy source here, happens again, and you have another energy source there. And this happens on both sides. Basically, you are creating with the help of the NADH, you are creating 4 ATP. Overall yield, and this is what I really want you to know, you use 2 ATP, synthesize 4 ATP, so you have a net gain of 2, and you also synthesize 2 molecules of NADH, and you split the glucose. Right, so we got 2 ATP and 2 NADH out of this glycolysis. What happens to the pyruvate end product? Under anaerobic conditions, lack of oxygen, it is reduced to lactate. This is why when you work out, you have what we call lactic acid buildup, which causes soreness in your muscles. So recently formed NADH transfers electron to two pyruvate molecules, reduces it, and becomes lactate molecules. But under aerobic conditions, it moves into the mitochondria and is oxidized. That means it loses further electrons. The second step of glucose metabolism, citric acid cycle. Pyruvate loses carbon atom, decarboxylation to yield acetate and carbon dioxide, exits in the bloodstream for elimination from body by lungs. This is where we get carbon dioxide from. Acetate is oxidized by NAD, linked to large coenzyme molecule to make acetyl-CoA and NADH. Two newly formed acetyl-CoA molecules enter your citric acid cycle. All right, so pyruvate, anaerobic, lack of oxygen, it gets reduced into lactate plus an NAD that got oxidized. In the presence of oxygen, enters your mitochondria. It's going to combine, lose a carbon, create carbon dioxide. That acetate react with an NAD and an acetylcholine to make this acetyl-CoA plus an NADH, which then go into your citric acid cycle. Now what happens in part two? So we've from glycolysis, we're able to get 2 ATP, net 2 ATP. Now we go into the citric acid cycle. Here we have 8 reactions, and this takes place in the mitochondria. So they do not require oxygen directly, but require adequate oxygen concentrations to proceed. Small amount of ATP is produced by substrate level phosphorylation. Adding a phosphorus group again. Here we go. And again, you don't need to know all the steps, but you need to know the results, basically. So you start off with this acetyl-CoA, combines to form the citrate and CoA. You have an oxidation that takes place. Then we start ATP synthesis. Then we have another oxidation that takes place. Each glucose molecule produces 2 acetyl-CoA molecules, which cycle twice, and your yield is 10 NADH molecules. These are electron carriers, basically, two FADH2 molecules, and 4 ATP, 2 from glycolysis, and 2 more ATP from the citric acid cycle. Little note here at this point, only a little energy has been generated, but you've conserved energy by transferring electrons to this NADH and FADH2, and that's going to go to your electron transport chain. All right, so this is the citric acid cycle. Citric acid cycle. So for each glucose broken down, this cycle turns twice. This is what happens. Now we get into the electron transport chain. Oxidative phosphorylation. This is where most of the energy is made, in the presence of oxygen, aerobic. Basic idea here is transfer of electrons. Again, generation and maintenance of hydrogen ion concentration gradient, and the use of the gradient to drive what we want. The goal is right here, synthesis of ATP. More than 15 electron carriers make up this chain, combined with 4 large enzyme complexes. Remember, enzymes are the catalysts embedded in your mitochondria. Electrons are passed down the chain between carriers in the form of hydrogen atoms. Right? Remember, hydrogen, the atomic number of hydrogen is 1. 1 proton, 1 electron. So we have these, what we call hydrogen atoms. This is how we pass down electrons. Right? The NADH and the FADH2 are oxidized. That means they lose their electrons. The flow of the electrons generates electromotive force. That's where we get the energy from. And that uses this force is used to pump hydrogen ions into the intermembrane space against their concentration gradient. Electron flow through complexes 3 and 4 continues to drive pumping of hydrogen ions. Complex 4 transfers electrons to oxygen. The final electron acceptor forms H2O, hydrogen from these electrons moving, oxygen from the oxygen that we breathe. The reason why we breathe, otherwise glucose metabolism would stop like glycolysis. We need that oxygen to be the final electron acceptor. This hydrogen comes originally from these NADH, FADH2 the electrons that are transferred down the chain, eventually binds with oxygen. Oxygen receives that, forms H2O. That's the oxygen that we breathe it in. Now we form water. It accepts that electrons. You have that transfer, that energy. Otherwise, it would be glycolysis, and we would not be able to make enough energy. Sometimes this is easier to look at in an actual diagram. So inside the mitochondria, NADH, FADH2, you have hydrogen ions, electrons being transferred down the chain, ultimately creating ATP and eventually combining with oxygen to make, as you see right here, to make H2O. Oxygen is the final electron acceptor, so that if you start at the top, basically the electrons are coming down the stairs. They need that oxygen at the bottom to bind with to make H2O to finish off the process. That's why we have to breathe oxygen. Simple slide, just putting that all together. Why do we breathe? People think that we breathe in oxygen, that oxygen is converted into carbon dioxide. Not exactly. Oxygen accepts the final electrons in electron transport chain. So as the FADH2 and the NADH become oxidized, lose electrons, hydrogen ions, eventually they finish off, they travel down electron transport chain, bind with oxygen, form H2O, and that's why we need oxygen in our bodies to finish off the reaction. That's why we breathe in oxygen. Oxygen that we inhale is picked up by the bloodstream, delivered to our cells, ends up in our inner mitochondrial membrane, accepts final electrons, becomes water molecule. Carbon dioxide is from the pyruvate, remember? When it enters the citric acid cycle, it loses one of its carbons. molecule of carbon dioxide and carbons are removed during the citric acid cycle. And therefore, that's where the carbon dioxide comes that we breathe out. It comes from the glucose that has been split into the pyruvate, but then before that pyruvate enters into the citric acid cycle, loses a carbon, forms carbon dioxide, and that's where we breathe out carbon dioxide. So they're related, but we're not breathing in oxygen in your body, converting that oxygen to carbon dioxide. It's two separate paths that lead to the carbon dioxide that we breathe out, or that lead to the oxygen that we breathe in, even though they're both related to our body's energy sources. Oxygen is very important, and this is just talking about it a little bit more on this slide here. The fact that oxygen is our final electron acceptor is a big deal. Try to hold your breath. You can't last long without oxygen. Same thing with carbon dioxide, very important. And if too much carbon dioxide were to accumulate in your blood, it would lower your pH, making it more acidic. And that could cause major problems as well. So your body needs to remove the carbon dioxide constantly and needs to continue to breathe in oxygen. Your body's always functioning. All right. Humans generate huge amounts of ATP per day, about 132 pounds. It's a lot of energy. So that's why we're constantly creating carbon dioxide and constantly using oxygen. That's why we need to breathe at all times. But this ATP helps power our organs, our digestion, our going to the bathroom, all different activities that our cells do. All right, and then how do we get to ATP synthesis? So the hydrogen ions have been pumped from the mitochondrial matrix into the intermembrane space, creating this concentration gradient. And we have this, we use this to create ATP. So the potential energy becomes kinetic energy. That does work when the hydrogen ions flow through the ion channels across this membrane. The potential energy becomes kinetic energy. It does work. Hydrogen ions flow through the ion channels across this membrane, and then you have the flow of hydrogen back into the mitochondrial matrix. Down the electrochemical gradient drives work of the ATP synthesis. So ATP synthase, that sounds like an enzyme, large protein in your mitochondria that functions as an ion channel, turbine, rotor, and an enzyme, catalyzes ATP production. What you wanted to do is ATP and that phosphorus group bind to ATP synthase to form ATP. So the ADP, adenosine diphosphate, two phosphorus and another phosphorus group, another phosphorus bind to this ATP synthase, this enzyme, and you form ATP. Hydrogen ions flow through the ion channel, and that's what those hydrogen ions flowing, those electrons flowing, gives it sort of like the electricity to make this happen. You saw that in that previous diagram, kind of showing it to you there. So how many ATP do we get from oxidative catabolism? This is what I really want you to know. So from glycolysis, 2, citric acid cycle, 2, energy release from oxidation reduction reactions of one of the NADH molecule pumps, enough hydrogen ions in intermembrane space to drive release of 3 ATP molecules. The FADH2 is slightly lower at 2 ATP molecules, but when one molecule of glucose is completely oxidized, the overall gain is 38 ATP. All right. 2 from glycolysis, 2 from the citric acid, and then from the oxidation of pyruvate to NADH to the NADH and the FADH2, each make their ATP, and then we have a total of 38 ATP from, remember, this is all from one molecule of glucose. And this is the big picture: oxidative glucose catabolism and oxidative phosphorylation. You may have heard of cyanide. It's very deadly. I think Hitler used cyanide to end his life, I believe. If anyone out there is Jewish and I am wrong, feel free to send me an email and correct me, but I believe that's what he used to end his life when he thought that I guess he saw that the war was going to be over and they were going to lose, which we are glad they lost. But cyanide, the reason why it's so poisonous is that it binds to the key enzyme of the electron transport chain and inactivates it. So your cells cannot produce ATP, even when oxygen levels are what they're supposed to be. Cyanide is very fatal. If your body, it's basically like if you stopped breathing. It's the same idea. If you stop breathing, you can't produce ATP. Your body will basically shut down and you'll die very quickly. Or if you take cyanide, which blocks the enzyme which produces ATP, your body will shut down and die pretty quickly as well. So now let's go into the fatty acid and amino acid catabolism. Cells are capable of breaking down fatty acids and amino acids. Remember, we talked about glucose. And this is it becomes very similar. All three nutrients are converted into chemicals that can be oxidized. Remember, oxidized, losing electrons, and electrons transferred to NADH and FADH2. Electrons removed from both fatty acids and amino acids are sent to the electron transport chain for oxidative phosphorylation, and it's very similar. All right, so most fats exist as triglycerides. Remember, glycerol with the 3 fatty acid chains. You remember that? Hopefully, you can review that earlier on in the book. Lipolysis, enzyme catalyzed process that liberates the fatty acids and the glycerols. Right, so we have both fatty acids and glycerol can be used for energy generation, but overwhelming amount of energy in triglycerides is derived from the fatty acid beta oxidation, which we're going to talk about. So the glycerol is converted into the glyceraldehyde 3-phosphate and enters glycolysis. Okay. Glycolysis, breaking apart of glucose. The fatty acids, and this is fat. Remember, fat has a lot more energy than glucose. Fatty acids are catabolized. Acetyl-CoA, beta oxidation, and into products called ketone bodies by ketogenesis. You may have heard of like these keto diets. This is, you may learn something about that very soon. But that's a very popular diet, and very much because of the science behind ketone bodies. All right, so the glycerol portion of the triglyceride goes through glycolysis. We know that. It's basically very similar to what happens to glucose. The fatty acid chains go through this beta oxidation. So in cells that can oxidize fatty acids, skeletal muscle fibers, and cardiac myocytes. Ooh, that's interesting. Skeletal muscle fibers. Is this why they say to exercise to burn fat? Fatty acids enter mitochondrial matrix. Fatty acid is bound to the coenzyme A, initiates a series of reactions called beta oxidation. The fatty acid chain is oxidized. That means removing electrons, and has 2 carbons removed. You get the NADH, FADH, acetyl-CoA, and shorter fatty acid with 2 fewer carbons. This NADH, FADH2 proceed to the electron transport chain, while the acetyl-CoA enters citric acid cycle. Another CoA molecule binds to shortened fatty acid, and rounds of beta oxidation continue. So it keeps breaking it down until entire fatty acid has been converted to acetyl-CoA. All right, so fatty acids can be converted into greater amounts of ATP. So you can see, depending on how long that chain is, remember those fatty acid chains are just long chains of carbon molecules. So whereas with glucose, we just had 6 carbons, of fatty acid chains, we have these long chains. And so every 2 carbons, we get an NADH, an FADH2, and an acetyl-CoA for every 2 carbons in that chain. So if that chain has 20 carbons, then we're getting way more electron carriers total than we would with 1 glucose molecule. Right? So this is just an example of a 6-carbon fatty acid, and it's going to keep producing these electron carriers, FADH2, NADH, and acetyl-CoA bound to 2 of the carbons, and then you have 4 carbons, and it's going to keep, you're going to go through another round of the beta oxidation. So it keeps going, cycling back through until you get all the way down to 2 carbons left, and that'll be the last time it goes through. And you just keep producing the FADH2 and the NADH. So what is ketogenesis? Process that occurs primarily in liver cells that can utilize beta oxidation. So it occurs in the mitochondrial matrix. 2 acetyl-CoA molecules are linked together. Acetyl-CoA, right here, that molecule, are linked together under certain conditions to form 1 4-carbon molecule called a ketone. Ketone bodies cannot be further metabolized by liver cells to an appreciable amount into bloodstream, where other cells can potentially use them for energy. Right, so we take this, breaking down this fat, form this ketone body, but it can't be metabolized by the liver, so it enters the bloodstream where it can be used by other cells. In other cells, ketone body can be split back into 2 acetyl-CoA and then oxidized in the citric acid cycle. So skeletal muscle fibers and cardiac muscle cells can use ketone bodies readily. Cells in the brain only use them during periods of starvation. Your brain normally uses glucose, not the ketones, but it can use them under certain conditions. During extreme caloric restriction, it sounds like a diet. Extreme caloric restriction, carbohydrate restriction, no carb diet, or full starvation, liver rapidly oxidizes, removes electrons from fatty acids for energy, leads to production of large quantities of ketone bodies. So I reduce this is why this keto diet, I reduce my calories or reduce my if I reduce my carbohydrates, body doesn't have enough carbs. What happens is your liver will oxidize fatty acids, basically burn fat, and produce large quantities of these ketone bodies. So it'll break down fat, or what we may call burning fat, break down fat, produce ketone bodies, which float around in your bloodstream and can be used primarily by your heart and your muscles, but also by the brain. Now, accumulation of ketone bodies in the blood is ketosis, can lead to dangerous lowering of the blood pH. So you have to be careful. there's always a downside to these to these diets. Scientifically, keto diets work, but because it causes your body to basically burn more fat. These low carb diets, you have less carbs in your body will burn more, will burn more fat. Fat is its sort of secondary energy source. All right. So and the goal is you'll lose weight. You don't want to do this forever. Right? You have to, it can be bad for your health, as you see here. Though, what is lowering your blood pH do? Well, then now, if you lower your blood pH, remember your body's pH needs to be at a certain, your body needs to be at a certain pH so that certain reactions in your body can take place. If you don't have, if your body's pH lowers too much, it becomes what we call acidosis, and this is called ketoacidosis. Then it can cause some reactions in your body not to happen that should be happening, and it can cause all kinds of trouble. Right? So you have to be careful with diets like this. Amino acids, dietary proteins are broken down to amino acid subunits in their digestive tract, and we talked about that. Absorbed into the bloodstream, delivered to your liver. Talked about the breaking down of protein happening starting in the stomach. We talked about that in the digestive system last chapter. Amino acids consist of carbon skeleton bound to nitrogen containing amino group. The nitrogen is not used for energy. So the actual amino group, the NH3, the amino group, is removed, and the remaining carbon skeleton can be oxidized. Electrons removed for fuel. These are the events that happen. Transamination, the amino group is removed and transferred to an alpha ketoglutarate. Generates two products, a carbon skeleton and an amino acid glutamate. The carbon skeleton can be converted into a variety of compounds, which then are oxidized. In the mitochondria of the hepatocytes, that's in your liver, the glutamate undergoes oxidative deamination, producing ammonia and this ketoglutarate. So some amino acids, amino groups removed as ammonia, are used for synthesis of new amino acids. So you can use this to make new amino acids when that amine group, that NH2 group is removed. The remaining ammonia molecules are removed by urea cycle. That urea from that broken down amino acid forms urea, then is eliminated by the kidneys, urine, and urea is forming two amino molecules. Ammonia molecules are combined with carbon dioxide. All right, so some of it is, when we break down amino acids, some of it is excreted into our urine, combined with carbon dioxide, made urea. All right, and that's the drawing of amino acid. And this is really a third. Your body is going to primarily use carbohydrates. A ton of energy from can be gained from fat, from triglycerides, from lipids, but then the tertiary option would be amino acids. All right, so this is just the big picture, nutrient catabolism, lipids, beta oxidation. Remember, 2 carbons at a time. Glycolysis, we take the 6 carbon carbohydrates, we break it down to glucose, 6 carbon sugar, then that's split into 2, 3 carbon molecules, pyruvate. In the presence of oxygen, it enters the citric acid and cycle and electron transport chain. And then proteins go through transamination. You make urea from the excess ammonia. You make urea, and the rest goes into the mitochondria. The carbons go into the mitochondria for and can go into the citric acid cycle as well.